Phelps, who is the director of the Minnesota Aquatic Invasive Species Research Center and a member of the faculty in Fisheries, Wildlife, and Conservation Biology. He is going up for one of the 2 promotions that professors ever get in their career for a full professor, and this lecture is his promotion lecture. Topic appropriate for the season: spooky stories and fish health and aquatic invasive species. Gives us a chance to see what Nick does. And I hope some of you, we put out an invitation for pizza after Nick's presentation in 100 Skoke. If you RSVP, totally welcome. And if you want to come and you didn't RSVP, I ordered for 8 extra people, so you probably can come in, no problem. All right, thank you, Nick.
Thank you. All right, and thanks everybody for being here today, sitting through the talk so you get pizza at the end. And Rob, thanks for providing that. For the folks online that can't see the room, everybody here is dressed up in appropriate Halloween attire. At least one person is. I appreciate the spirit. Yeah, so as Rob said, this is a promotion talk of sorts. My goal here is to just give you a sense of what I do. And I tried to pick a few stories here that highlight sort of the diversity, the breadth of the things that I work on. Likely some things that you don't know already. Oops. So for those of you who have not been in a, they're not moving forward. There we go.
Saying screen share, you started your screen share.
It's not seeing anything?
That's not seeing anything.
Okay, let's see what we got. How's it looking, Chris?
Looks good. I'm seeing your speakers.
No, you all see my notes. Where'd I go?
Yeah.
How do I swap screens now? I don't see any of my. I came early to figure this out. What do I do, Pils?
You try and exit out and swap screens. Is there a search for like swap screens? Can you try and share it now? Is this what you had before?
Mm-hmm. And then I don't have a swap screen button anymore.
You know where that is?
Where is it?
It's in the top right, but right in the top, maybe.
We're not nuts. Oh, it's over there.
Oh, you can't see that.
I can't see the. There's.
Yeah, there you go.
Now it's. Does anyone have a way around this? So now it's on the Zoom, it is showing that you swap one way and you get back to the other way.
I know this and I know that. Okay. Oh. I'm just.
Oh, you can't find my mouse over here.
Oh, got it. Okay. So new share.
Thank you, Cassie.
I'm gonna try. Hang on. Portion of screen. Just wait.
Gosh.
I know.
Oh my God.
You're welcome.
Yay.
Good?
Okay. Thank you for being here today, coming for the pizza. Solomon, you haven't missed anything. Okay, we're all caught up. For those of you who have not been a part of a promotion process here, this is basically what it looks like. You got the dean and the department head asking you, what do you do here? So for all of your references, it's coming up very slowly for some reason. Okay. All right, so what I do, I study emerging threats to aquatic systems at the macro and microbial scales. And I'll give you a sense of that here coming up in the fields of fish health and aquatic invasive species. It sounds, when you see these projects, I'm often jumping around on topics, or it seems like they're not connected, but really what I'm trying to do is solve problems, identify the threat, figure out what the risks are, and come up with management plans, very applied, focused management plans on how to address those problems. One thing that is consistent in the way I approach these problems, sort of the scale and the sort of the scope or approach, some might consider it a philosophy, where you incorporate aspects of the human system, the environmental system, and the animals that live in it, all together. It's a complex, connected, and dynamic world, and when you live in just one of these circles, you're undoubtedly missing important things. When I was in the academic health sciences, I was in the veterinary school for 10 years, we would call it One Health. We rebranded it as ecosystem health. It's a friendlier term to us up on the hill, sometimes called eco health, but it's really that intersection between animals, humans, and the environment. And you'll see that through the examples I give you here in just a minute. One thing that this approach requires is a highly interdisciplinary approach with people from across disciplines, people on campus and in the real world, locally and around the world. This is really team science at its core, and something I really like to do. But I do want to acknowledge the many people and organizations that I've worked with, but highlight the grad students that have been in my lab or worked closely with it. This is the best part of my job, I consider. You can see those that have gone on are changing the world, and those in the red are here right now and will change the world someday. So just to give a shout out to the students that have been a part of this. And you'll see as you sort of scan around real quickly here, we have some in conservation sciences, water resource sciences that we're all familiar with, but also veterinary medicine and professional students in the DVM program and public health and masters in public health through the veterinary program. So this really interdisciplinary approach is the way I've tried to build these teams. And you can see that in the students that I've supported. All right, so that's the overview. You're here for the spooky stories. I'm going to leave the blood and gore, the rated R version, for after hours. I'm going to focus on some examples that are particularly complicated, sometimes controversial, and to many, a little scary. They're all projects that are series of projects that have kept me up at night anyways. And since I always have way too much to say and the amount of time that I have to say it, we're going to have this be a bit of a choose your own adventure, because we may not get to all 4 stories. Depends on how fast I talk. So I'll open it up to the folks in the room. If there's one that you want me to start at, we'll go there, and then we'll see how much we can get done. So, brave person, maybe somebody with a fancy hat in the back. You want to pick one? Number 1? Okay, that's easy. All right, so attack of the killer viruses. The more you look, the more you find. So my background, just very briefly, I got my PhD in the veterinary school here on campus. My master's was in aquaculture and fisheries, but really a focus on fish pathology. Fish health is really at the core of what I've done a lot of, and this is a little highlight into that world. What are you seeing? There we go. All right. So fish get sick too. Scary. This is a parasite I described or characterized a few years ago, liquefies the muscle of fish. that are important here in Minnesota and the Great Lakes region. We've worked on bacteria that'll eat their way right into the muscle of fish and down to the bone in some cases. If I'm known for anything in the fish world, fish health world, it's viruses. I have a fair bit of background and effort in the past, focused on virology. And fish get a lot of viruses, as I'll show you here in a little bit. We use a variety of methods to work with and to understand and identify viruses. Histopathology, where you embed tissue into paraffin, and you cut thin sections of it and look at it under microscopes. You can see the structural anatomy, the ultrastructural anatomy is a lot of pathology is observed that way. Cell culture, where we grow the viruses in the lab on plates, and these culture plates where they have cells and you intentionally infect the cells. Electron microscopy, there's a variety of ways to do it there, but with this approach, you can actually see the morphology of a viral particle and it narrows down what you're looking at. If you don't have any idea what's growing, EM is the route to take. And a big focus has been, in the direction that my lab has been going, is on molecular approaches to virus diagnostics. We use traditional gel-based PCRs, we use real-time quantitative methods, and now most often next-gen sequencing, where we're amplifying everything, the whole virome of the sample, and seeing what's out there. Now, this is really helpful if you have no idea what the cause of the problem is. So we go out to fish kills, mortality events that happen in lakes and rivers, and we get those samples and we take them back to the lab and we just start sequencing things. This is an example that will come in a second here of a tiger muskie that we got in Lake Nokomis. It washed up dead. A big tiger muskie is kind of alarming to the crowd, so they called the DNR. The DNR sends us a sample, and we found a whole variety of viruses in there. Like any other animal, like us, we've got viruses in us now that are just hanging out, doing their thing, not causing problems, and most of these probably are. What we look for with this approach are those that might be related or novel in certain families that can cause major problems, known problems in fish. So reoviruses could definitely cause problems. The parvoviruses, maybe circoviruses, we find a lot, and some of those we are understanding to be highly pathogenic. The coronaviruses too. So this is that first step. I've got myself my haystack now, and I can start picking off one at a time and diving in and figuring out which one may have been most likely to be associated with the disease, if any of them. It could have been a bacteria or parasite or water quality too, but for this approach, we've been looking at viruses. As we increase our sample sizes and the numbers, we've been working a lot on common carp. These are from 5 populations of common carp that we've sampled, all healthy fish. We go out there and we just set nets, get what we get, sequence the fish, and you can see the huge variety of viruses that are in those samples. A lot of viral diversity. Many of these families have never been found in fish before. We don't know what they mean, but they're out there, and it's definitely worth looking into. And as we learn these things, we start picking off certain ones to understand better and what the risks are to populations in the landscape. Now, targeting the sick ones for common carp, for example, we looked at 22 different mortality events of common carp over a couple-year period, mostly here in Minnesota, but elsewhere in the Midwest to Pennsylvania and Iowa. A lot more viruses. You can see the difference compared to that previous slide. And the two that really jump out at us, there's a delay, sorry, are the pox virus and the alloherpes family. Those we know can cause major disease problems in carp, and since we're seeing them in our mortality events locally, this was an indication us to dive deeper. And at the same time, this was making headlines. There's a lot of local newspapers that were picking up on this. When 10,000 carp wash up on the shore over a 3-4 day period, the public takes notice and they call the DNR, and then the media gets involved. And so we were seeing viruses out there. Our lab confirmed koi herpes virus for the first time in Minnesota. And we also found another one that I'll show you here in just a second. So koi herpes virus, also called cyprinid herpes virus 3, it's the technical term, causes gill necrosis, hemorrhaging in the skin and throughout the body. these fish die in high numbers, potentially once infected, naive individuals. This is carp edema virus. The other one, this is a pox virus, a big problem in carp aquaculture. This video is super choppy, so I apologize for that. But what you hopefully can see in the still images for sure, this is a virus that causes the skin to essentially slough off. It's losing its skin, the gills are getting necrotic, they can't breathe, they're suffocating down there, highly pathogenic. This one is a bit more concerning than KHV because we think that this one might be jumping hosts in different ways. So we went out and looked with that early indication from the next-gen sequencing. We went out and targeted populations across the state. Please come up, slide. For both these viruses. Do you see it online, Chris? Okay, everyone online's getting approved here. There we go, I'll just skip ahead. So this is Isaiah Cholo. He was a grad student in my lab. He is now the head of the pathology lab at the Minnesota DNR. He went out and found all these locations. We put the first dot on the map, and within months, we had 20-some dots on the map. The more we looked, the more we found, and that created a big sort of stir in the fish health world of, is this something that's emerging? Is it spreading? Did it come from somewhere? What does this mean? And now when you look at these global maps of this internationally reportable viruses, both of them are internationally reportable, Minnesota is this hotspot. Probably not because it's breaking out here or something, but just because we're looking for it. So it opened up a lot of management questions. One of them was about host susceptibility. How concerned should we be as good Minnesotans about our walleye, as all of these viruses are floating around? And for KHV, we looked at this one pretty intensively. We found it at about a 20% prevalence in a carp population, cohabitating fish from that same location. Nothing. Highly specific to the carp, not really a risk to anything else. We focused on the buffalo that cohabitate. They live right together, similar life histories as common carp in this system. They're nose to nose, and we know the virus transmits that way. And they were all negative. So highly species-specific virus in this case. I've got a lot more that I could say about KHV. We looked at the transmission and behavior changes once infected in the lab. We looked at seasonal dynamics and wild populations that have been exposed, age class susceptibility. We, for the first time, characterized the disease periods of how long are they infected before they get infectious, when do they get sick? So essentially, when do the fish get the sniffles? That's important information as we try to model this disease in the wild. What's it going to do if it gets out there? How fast is it going to spread? The same modeling approaches that we needed when SARS-CoV-2, when COVID was spreading. These are all boxes people had to check for that virus, and we're doing the exact same thing here for KHV. One of the big questions that folks had was, where did it come from? For the sake of time, I'll just skip forward and give you our two hypotheses. The first one, it was first found in koi, hence the name koi herpes virus. Because koi is such a big industry, a hobby industry, there's huge value in the koi fish. Same species as the common carp. There we go. And so that's where they looked first. Sick koi, get attention, common carp, do not. So these were our two culprits. Either it emerged in the koi somehow in the 90s and spread throughout the world really fast through the koi trade, or common carp were the host, and this has been a virus that's been with them their entire life and was imported into the United States when carp were back in the late 1800s. We did a whole series of molecular projects. No, it's really wonky here. I've got so many screens going here. Okay. We did a series of molecular projects, which, long story short, suggests that the virus co-evolved with the carp. And when they were imported from Europe, they brought with them KHV. And the KHV later spread to koi through sharing of water and interbreeding and things like that. The genetic lineages, the mitochondrial genes of carp overlap perfectly with the variable nucleotide tandem repeats of the virus. So we see a lot of overlap there, and that suggests that this has been here a long time. Now, the killer virus part and what we do about it, this might surprise you, but here in Minnesota, funded through MACERC, we've been looking at the possibility of releasing this virus. We don't like carp on the landscape. They cause huge ecological problems, and this could be one highly selective way of removing them, or at least suppressing the fish. So some of this work was featured in the New York Times as sort of this innovative approach to pest control. We've never done this in an aquatic system, although pathogens are used to control terrestrial pests and egg pests. It has never been attempted in an aquatic environment. So a fair bit of controversy. The Australian government has been funding for about a decade. They've invested millions of dollars into the use of koi herpes virus as a biocontrol strategy in Australia, where carp are a huge problem. They haven't yet released it, but they are getting awfully close. And it's highly, highly controversial over there. So, if you are scared of what I'm saying, if it comes off, I apologize. You're not alone. The idea of releasing viruses to control things is scary, especially post-pandemic. Our understanding of and concern is rightfully much higher than it was 5 years ago when we started all this work. I mean, it's playing out in the literature. There's high-profile papers on both sides of this issue, and our research has been informing this conversation. We're often getting excited, usually more on the favorable end of the spectrum, demonstrating the host specificity, the disease periods, and the likelihood of mortality. The fact that it's endemic, those types of things might lead it to more of a safe option for control than the scary one. Very different situation in Australia, where they don't have this virus and have a different situation to deal with. So that's my story on killer viruses and looking for them. Any other stories you want to hear about next? Three. Okay, too hot and nowhere to go. I mentioned before that these fish kills, I've been working on fish kills for a long time. I'm not alone. Across taxa and around the world, mortality events are becoming more common, and they're becoming bigger in scale. So more animals are dying, and they're dying more frequently. This is a huge problem. We've spent a lot of time trying to understand at our local scale what this means, just to get a sense of what we're dealing with here. So a while back, and this was when I was in the veterinary school, we created an online database. People can go into this portal. They can report fish kills if they see them. This is for the public, for managers, and they put dots on a map. It emails me and the DNR pathology lab right away. So we can triage these reports and decide if we need to go out and collect a sample. This is how I get a lot of the information for the fish kill work that we do. And there's a lot of data here that we can mine and look at. And if I was a betting person going to go to Vegas on a fish kill wire here, the most likely mortality event is probably going to be a sunfish or crappie in June due to warm water or columnaris, which is an opportunistic bacteria that's everywhere and likely observed in an urban area. That's the pattern we see. Oh, there's lots going on here. That's the most common. Now, the in June part piqued our interest. We know, and lots of people have been working on this idea that hot weather, the warm water causes stress to fish, and one possible consequence of that stress could be death, either directly from the water temperature or indirectly from secondary infections or low oxygen levels. This is always in the headlines in the newspaper. The DNR talks about it a lot. We heard a few weeks ago from our very own Gretchen Hansen about thermal optimal ranges, and as temperatures shift, what we're likely to see are these shifts to less ideal conditions, physiologically speaking, and causing stress and changes to populations. So you're not seeing the same things online and in person, but that was the point there. We looked at this, and in the past, our research would suggest that that's definitely true. We did some pretty basic modeling approaches looking at sort of predicting where fish kills might happen. The number two most likely cause that popped out in these models was the overnight air temperature as a driver for fish kills. The warmer the overnight air temp, the more likely you were to see fish kills happen. When we started digging, it was just an early indicator. There's a lot more to that story. So we partnered up with a team that had a whole bunch of data from Wisconsin fish kills. And we really hyper-focused on the idea of climate change, the water temperature increasing as we go forward, and what that means to these populations. So I'm not going to spend a ton of time going into the details of these methods, but I'll give you a couple punchlines here. And I highly regret putting animations into this talk now. All right, what you're not seeing in person or online. As we go forward into the future, two trends become really obvious. Number one, the number of fish kills, the predicted number of fish kills from these models go way up. Folks online can see that. For those of you in the room, you should, for some reason, it's just very faint, but you can see this line way up here in one. Spooky indeed. Okay. All right. I don't know, Chris, what you can see online anymore, what I have seen in person.
It's the predicting fish kills slide still.
Okay. All right. The second observation, in addition to the number of events going up, as we are seeing already globally, there's huge differences in the predicted number of fish kills when you consider air temperature models versus water temperature models. And Gretchen talked on this a few weeks ago about the important differences there and understanding those differences. Historically, everything was built off air models, air temperature, and there's likely a lot of value going towards the water temperature approach. All right, so now what some of you are seeing, so it's not just as simple, or our hypothesis isn't just as simple as the hotter it gets, the more likely you are to die across taxa. Now, this is a bit of a complicated figure, so I'm going to break it down here, hopefully for you. So across the x-axis here, we have different taxa of fish. To the right, as you're looking at it, warmer, tolerant fish. You've got the centrarchids, the ictalurids. They can tolerate warmer temperatures. And on the other end of the spectrum, we've got the salmonids and esacids that like cold water. This y-axis is the difference between their thermal optimum and the temperature at which we saw them die from fish kills. So we just took all of our fish kill data, put it into this thing, and see how far that difference is to see if patterns held up. But you'd expect, if you think it's warm water factor, that the higher the temperature, the more likely it is to see a fish kill. So you want to see stuff on the top of that line. Now, man, what you see online, oh, this is really too bad because this is an awesome figure. Okay, great. So what you see on the left-hand side of the screen is that pattern holds up for cold water fish. They do not like hot water, and when they get exposed to it, they're more likely to die. Compared to the other end of the spectrum where they might be able to tolerate some hotter temperatures, at least from a mortality. It's not going to cause them to die. It might cause stress, and it might cause reproductive issues, and it might cause other things, secondary infection, but it's not because of just the warm water. There's more going on here than just water temperature as you think about it across taxa. And I like this next, this is going to get a bit of a tangent and a little too scary, but man. All right, the folks online are seeing a news report from Arkansas, and I can poke a little fun. I spent a couple of years down there. They saw a big fish kill, the biggest fish kill they'd ever seen, freshwater drum. Coincidentally or not, UFOs flying over them. Anna Pfeffer, the world's foremost ichthyologist, which doesn't have any papers, you can correct me if I'm wrong, Cassie, also the Olympic badminton champion. She investigated this and found the mark of the alien on the fish. So there's a lot of things going on with fish kills. Temperature is one of them. And my lab is trying to understand these drivers to figure out ways we can deal with it. How do we move forward? Because fish likely feel like they're stuck in a gym with Carrie, right? There's nowhere they can go. They can't just walk up north or turn on the AC. So there's options I would argue that we ought to be focusing on. Perhaps assisted migration or assisted evolution. There's people with fisheries thinking about climate change. How can we genetically engineer populations to be more resilient to warmer temperatures? We can do that through formal genetic engineering where you're manipulating genomes or just selective breeding. The salmonid in the Pacific Northwest, that's a hot topic right now. Supporting site-specific or landscape resiliency, obviously important. Maintaining what we have as best we can. An alternative, though, is just adjusting our expectations. Maybe we need to move the goalposts. Maybe this is just the way it's going to be, and we need to change our view of it, and fish kills will just be part of our life. Now, that last idea might feel like we're letting ourselves off the hook, and I would totally agree with you. But this quote is from a newspaper article entitled "What You Need to Know About Fish Kills." And it was a Minnesota publication, high-profile one from a well-respected biologist, saying that maybe fish kills and what's causing them is really a good thing. It's culling the weaker individuals, more food for the others, more space. I've got a big problem with this statement, and I would argue that we need to do more. And my lab's been working on sort of the resiliency question and understanding the drivers so we can do more. All right, we got 2 left, about 20 minutes. 2. All right, this is my favorite story. So this has been a long-term interest of mine that I've been able to circle back to. And I'll start out big picture here. Bait fish, for those of you unaware or not local to Minnesota or engaged with the fishing industry, bait is a highly sought after product throughout the year for angling. It's important to the culture of Minnesotans. We've done surveys and suggest that 70% of anglers are using bait, and the vast majority of them say that it's very important to their fishing abilities, that they need to go out or they want to go out, get bait, and go use it to catch fish. It's also economically important. There's a multi-million-dollar recreational fishing industry. When we think about the 1 million+ registered or licensed anglers in the state, 70% of them wanting it, the multi-billion-dollar industry that is, there's a lot going on here. At the same time, what we're seeing online are some significant issues with that bait supply, both in their availability and the risks that they pose to the environment. So I've got a couple of snapshots that just highlight these issues, the shortages that are being experienced by the bait harvesters and suppliers, the bait shops, the anglers. They're all upset. It's really hard to get some species that they really want at the right times. And this has put that industry in many ways at odds with the DNR, who regulates the bait trade. In Minnesota, for example, you cannot import wild bait. You have to harvest it or grow it here in the state. And that's a protection. It's meant to reduce the importation of bad things, but you can get into these complicated messes between the dynamics of allowing an industry to thrive and mitigating risk. And that inherently has conflict. So folks in the room, there was more screenshots, cool ones, I promise you, that emphasize that point. So we've spent the last 5 years or so thinking about those risks, trying to put some context and data to the conversation in ways that we can move forward and be scientifically or provide evidence-based decision-making instead of just sort of perceptions and concerns on both sides and a lot of uncertainty. So a lot of this was led by Meg McCachran. She was in my lab, and this was the topic of her thesis. But you can see we went through this sort of expert solicitation process. to identify what might be out there, we went out and did surveys at bait shops, seeing what was actually out there. And then we looked at what appeared to be, at least our hypothesis at the time, the big driver of that problem, the recreational bait users, to understand their role in this situation and what opportunities we might have to reduce risk by targeting that population. So a lot going on here. This is a whole talk to itself, but one thing that had emerged from a lot of this work was the risk from this particular parasite called Ovipliostophora ovariae, most commonly referred to as just the ovarian parasite of golden shiners. It's hard to see in this image, it's a little blurry, but these are, that's the ovary, that sort of yellowish spot in the middle of that golden shiner up here, where the mouse is. And if you look at it in real life, what you can see are some of those eggs are white, they're opaque, they're dead, and others are still viable. It's because of this tiny little microscopic microsporidium. It's most closely related to a fungus. It's a spore that replicates inside of a host cell. It's operating intracellular, it's inside the host cell. And as it multiplies, that cell ruptures and dies. And the more that happens, the more damage it causes. And in this case, it will lead to sterility, sort of castration of female golden shiners, because their ovaries are just obliterated, the eggs are all dead because of this disease. So as the fish ages, it gets worse. We've known about this since the 1960s. It was when it was first described, coincidentally, at the same time that the golden shiner industry was growing. So like that koi carp thing before, we found it in the industry, probably because that's where we were looking first. That's where the money was, and that's where people focused their energy. There's been very little research in the last 60 years trying to understand the impacts to wild populations, the dynamics within populations. My master's thesis was on this particular parasite. And at that time, we didn't even know, I mean, my job at that time was to just figure out the life cycle. We didn't even know that, even though it had been essentially sterilizing these populations in these farms, multi-million-dollar industry. Nobody had cracked that nut, and we're still trying to answer important questions. Because of the concern and that uncertainty, there was a lot of management and reaction. Bait fish are being sent around the country. Some states, Minnesota, I mentioned, put a prohibition. We're not going to allow importation, in part because of this parasite. The state of Maine listed the ovarian parasite as 3 pathogens as justification for that state banning importation of bait. Taylor, this bait importer, he took the state of Maine and that rule all the way to the US Supreme Court as a violation of the Interstate Commerce Clause, that states can't sort of ban things from coming into their boundaries for market protection. You have to allow interstate trade of products. It's sort of this, we're a union, and that is something that they all want to abide by. Unless you can justify the risk that importation may pose. So they argued at the Supreme Court that this was a scary disease and it's something that ought to be regulated there. And it was upheld. You cannot import golden shiners into the state of Maine because of this parasite. In Minnesota, we have this prohibition, but we have a huge golden shiner industry locally. There had been no surveys of golden shiner production ponds. We didn't know where it was. We had not looked at wild populations of golden shiners. I did that survey of bait shops back in 2015, 16, and we found it was widespread at the bait shops. But the DNR, we had, it's a long story, this Operation Bucket List. There's this undercover operation with the DNR, sort of a sting op to catch people illegally importing bait. So there was intentional targeting of high-risk bait fish importation. And so their response to these results was, well, it was probably the imported ones where you're finding it. And they were operating under the management assumption that because we hadn't found it, parentheses, hadn't looked, it wasn't here, and we should continue these management practices. And there's been reports, the DNR commissioned a report, because this plays out at the legislature every year. Will once again this year, probably. This, should we import golden shiners from Arkansas story. And this big report that the legislature commissioned, the DNR led it, was Jeff Gunderson, who some of you may know, formerly of Minnesota Sea Grant. He had retired and did this on the side, but he found this ovarian parasite to be one of the main reasons why we should not import golden shiners. Under, again, this assumption that it's not here. So, obvious question, is it here? Now, going back to like what I do, I like to think about these emerging threats, find them, understand them, and then mitigate the risks of them. So this was an obvious place to start. The DNR didn't really want to know, to be honest, and so we went, we cooperated with the baitfish industry. They went out to wild populations and we went to their farms and we looked, and I will skip for the sake of time the methods here, but suffice it to say that we went to 16 locations. We got good statistically representative samples. We used a variety of methods, mostly molecular, to find them. We sequenced things when they were new. And the next slide will show you what we found. Just for the dramatic reveal, if you see blue, that's positive. So we found it everywhere. Every single location we looked, we found Ovipliostophora. There was one site that had very low level infections, and we did this through quantitative PCR so we can see how much DNA was in those samples. But it was nevertheless positive. It's hot. Every site is moving around. I know it is around the state. And this was the first conclusive evidence to show that. So yes, we can check that box that it's here. And the next management question is, okay, now it's here. How do we approach that? It was interesting, this conversation that transpired afterwards between the aquaculture industry and managers, both locally and in other states, and the fish health profession, what we do with this information, because they have pretty big implications. I mean, there's the Supreme Court thing and all the stuff that's out there, like legal implications of these results. So they held this up as, this is why we have prohibitions of bait. This is why we do fish health management, because we want to keep things out, and something must have snuck in. Like we busted these people for illegal importation, it must have been them. And now you've got liability being thrown on the table of this company that was illegally importing, may have introduced this to the state. The finger got pointed squarely at the state of Arkansas because that's where the biggest golden shiner market is. 80% of the golden shiners produced in the country are in the state of Arkansas. The farm I did a lot of my work on for my master's, they produced, just to give you a sense back then, something like 1.2 billion fish and sent them around the country. So no doubt they were likely importing them from Arkansas, and that finger could justifiably be pointed in that direction. Once again, mismatched from my screens. But barely, there you go. All right, so in my sort of annoying sleuthing that I can sometimes do, I found this industry newsletter from the mid-70s. This is like an egg industry newsletter for the folks, Arkansas, Missouri, Louisiana, like that region of the country, called The Rambling Along. It's this, it took some hunting to get this. But one piece that I will highlight was this quote, and it's not highlighted here, but the bigwig who sort of created the Arkansas golden shiner industry was credited for importing those original broodstock from Minnesota to Arkansas. Now, this was long before this whole controversy of Ovipliostophora was happening up here, and I found this little nugget. So I mentioned this in sort of a hallway conversation with our great Andrew Simons. How can I go back in time and figure out if those broodstock from Minnesota were like, which direction should this arrow be pointing? So Andrew and I got to chatting, and it turns out the Bell Museum is more spectacular than I even realized. We did some invasive sampling of the collection, and I got fish from Andrew dating back to 1893. 75 lots from across the state, not a lot of individuals per location, but enough mature females to do some diagnostics. And we went in and we aseptically collected everything. We were very, very careful given the implications here. We used molecular approaches and histopathology of both our target parasite and the housekeeping gene EF1 alpha, just to confirm that these samples hadn't been degraded as they were stuck in formalin and ethanol for decades, more than a century in some cases. And we confirmed everything we found by sequencing, just to be 110% sure we knew what we were doing. So the folks online are seeing it, you in person are not. See if I can. Okay, so dance back and forth. So what we found, 22 of the 75 lots that we tested were indeed positive for this parasite. I'll throw up some arrows here. This first one. Gosh, now I've got 3 different things going on. I apologize for the technical difficulties. So this arrow on the furthest right, that's when this parasite was described in the literature. That's when we found it on golden shiner farms. We found it all the way back to that very first sample in 1893 in broodstock from a wild population in Minnesota. The first time we were able to get both molecular and morphological confirmation to really be sure was in the mid-1940s. Regardless, before those fish were brought down to Arkansas. And here's just some of those pictures. These are eggs, these are yolk globules. The eggs with the stars, that sort of pinky shade in the middle, that's just solid spores. You look at it, these are all spores, just destroying that egg. So we know it's out there. Our conclusion at this point is that it's likely an endemic disease of golden shiners that was introduced to the Arkansas production system from wild Minnesota broodstock. And for whatever reason, it's a much bigger problem down there. If we compare the sort of quantification, the severity of disease in these different populations, it's way worse down in Arkansas and other warmer states than it is up here in Minnesota. So we have a whole series of questions that we need to answer before I can tell you the end of this story. I don't know what the management recommendations are yet, because I don't know how bad it's going to be on wild fish populations. I don't know how bad it's going to be if this is a temperature-driven issue. And the hotter the temperature gets, like it is in Arkansas, that's just going to make it worse here in Minnesota. There's a lot going on. Disease ecology is like a fascinating place to work, and that's where this whole line of research is going. So stay tuned on this one, maybe a subject of a future talk. And I am out of time. I'm going to save the last 10 minutes for Q&A if we have it. If you're all quiet, I can talk to you about the runaway invasions. But for now, let's just go to the end. Hey, Dan.
So if you look for viruses in dead fish and you find them from like 10 different viral families, how do you decide what is the positive virus for them?
Okay, so for the folks online, Dan's wondering how you can tell the cause of a disease when you get a whole suite of potential viruses from sequencing. So the way we approach that, we have to fulfill Koch's postulates. You have to be able to isolate something and then be able to grow that and then reinfect that animal and see the same clinical signs and re-isolate it. So you have to be able to complete a cycle. All we're seeing with that first, those first identifications and the spreadsheets I showed, is that they're in there. We do not know that they cause disease. We can't grow most of them. A small fraction of viruses can be grown in the lab, and so you can't do that. What we sometimes have to do is homogenize the tissue and then direct inject the homogenization in there and see if we can get the virus to take root. So many of them we don't know. There are a few viral families that are easy to grow, we know cause disease, and so we'll often focus on those first. But there is a huge world, the virome of animals is growing. You'll be hearing about the viromes like you heard about or hear about microbiomes containers. Like the viromes of people and animals are important, and that we're just starting to crack the surface here on fish. Yeah, Sushma.
This was great, Nick. I really appreciate knowing what you do. and, learning all the different things to do. I'm actually, I can ask a very like basic logistics question, like, where are you doing this? Where's your lab? I'm figuring out what our colleagues are doing. I don't even know where parts of the WCB do it. So is this in the vet school or is this in the, I'm assuming not in scope?
Yeah, so the question is, where do I do this? So I've, in the spirit of team science, I've tried to leverage available resources everywhere. And I've got little boxes and freezers all over the place. Trying to not only maintain collaborations and partnerships with the individuals that work in all those places, but because it requires a lot of different types of stuff, expensive stuff. The diagnostic equipment is insanely expensive. So I have a benchtop lab in Hudson, in the 3rd floor. It's got a giant Lorax on the wall if you ever want to come and see it. It's a cool space that we renovated for like fish health diagnostic work. For the fancy, more analytical equipment, I partner with the veterinary diagnostic lab, and that's where my office used to be before I came to FWCB. So I work a lot with the folks down there, and I route a lot of samples through them and leverage their expertise a lot. I also work with the DNR's pathology lab. There's a big fish health lab down in La Crosse, Wisconsin, managed by the U.S. Fish and Wildlife Service. We all have our specialties, and we branch out in that way. But most often you'll find me and my folks either in the vet school or in Hudson Hall.
And that's where you're doing the expert.
I have got the PCR-based diagnostics I can do here. The histopathology, I can read slides here, I can do that stuff. The culture-based assays are all done in the vet school or the DNR. Yeah, Joseph.
Well, first, I really hope you started writing your popular science book, Operation Bucket List. It's a fantastic story that you bring a lot of things. I'm serious about that, and you tell it well. It seems like someone would naturally propose you could label fish that are grown in aquaculture and be able to trace if there is smuggling, and I'm sure the industry would be resistant to that. Could you comment on whether that's been proposed? I'm thinking like stable isotopes or DC markers that you could treat them with in a pond system so that if you did sampling, you could see if they're showing up at certain places.
Yeah, great question. So if you didn't hear online, the question was about marking individual bait fish to see, check their origins. And that's something that's been discussed a lot. It's impossible right now to track them. And I was on this USDA task force meeting back, I don't know, 10 years ago now. And they had all these like swine and cow vets there trying to figure out how we can track our animals. And ear tags, like, yeah, fish don't have ears. What about tail tags? Like, yeah, they're this long. Like all these like bizarre ideas. They have not figured chemically, that's where it was going. There was a lot of concern for the types of chemicals and labeling and how that might be introduced like intentionally into food fish. There was concerns about just the scale of it and the uniqueness, and how could one farm have its own variety of marker versus another farm.
Sure.
Because it's the commingling that we really get concerned about, wild and farm together, which makes us nervous. So there's been thought in that direction. What they've done, what Operation Bucket List did, is they essentially, but the way they described it to me, is essentially sandblast the fish with these fluorescent particles that would embed right beneath the skin and look normal otherwise, but you put them under UV light and they glowed. So they could walk in to these bait shops, flash their lights, and you got like a glowing tank. And you know that came across the South Dakota border. Because they did it on one lot. They knew exactly where they came from. So there's ideas like that, more on the enforcement realm than in practice for aquaculture, but yeah.
On the climate change story, it's really interesting what you said about this evolution. I'm just curious, like, my mind immediately went to if you were to do that, and then it becomes specific and first, and then like how other conversations I think about like how that would affect the system.
Yeah, so the assisted evolution of species to adapt to whatever you want it is them to adapt to, isn't a new concept. It is species specific generally, or gene specific, depending on what you're targeting. And there are, depending on what you do and how you manipulate them, it could have big implications for the rest of the environment, some of which could perpetuate across generations and surprise us in weird ways. We've been assisted evolutioning aquaculture animals for decades, crossbreeding, like natural crossbreeding to select for cool color varieties in goldfish and koi, or disease resistance in tilapia and trout. The idea of using it for ecosystem response is still relatively new. It would be like, could you harvest broodstock walleye from southern Minnesota and continue to breed them, increasing water temperature in the tank and only taking off the healthiest ones from the top? And over time, you're going to get a population that can tolerate warmer water. Now, one might argue that's great and makes the system more resilient, and you haven't done anything unnatural because you're just sort of speeding up the process, what might otherwise happen in nature. Another way to do that is to find the gene responsible for thermal tolerance, cut it out, crank it up, do whatever you want to it, and pass it on, heritable trait, through CRISPR-based engineering, and the world will never be the same. Now, one might argue that that's the same thing to the same end game. One's more or less natural and one's in a lab. The consequences of which are probably pretty similar, regardless. So it's a fascinating thing. There's lots of sort of ethic papers that are coming out on this right now. The conversation is the hottest in the Pacific Northwest with the salmonids, because there's whole runs that are collapsing because the stream temperature, which is so critical to their success, is changing year to year, and they're just losing entire runs of fish. And that, there's no coming back from that. And the cultural importance is like no other. And how far do you go? That's it would be a great debate conversation over pizza.
Yeah. Question on.
Oh, good. Oh boy, a bunch of them. Which one should I tackle? Okay. All right, so we have a question online about a logistical question about the sequencing. They have a field ecologist background, and they're trying to use this in their research now. Did you identify the sequences, or who do you look for to ID? What can you tell me about describing the certainty of identification? Okay, so the sequencing methods, we've used a couple different sequencing approaches. We're in, first there was like Sanger sequencing, where you identify each base from a targeted sequence. That'll tell you what's in this particular short sequence of interest. That's how they constructed the human genome. It's just all these little short sequences all stacked on each other. The next gen sequencing is where you sequence all the DNA or RNA in a sample, chop it all up, and then computationally realign them and find overlaps in those segments, and you stack them up. It's insanely computationally intensive. This is what MSI was built for at the university, was at first to deal with the genomic data that the genomic center was generating. Huge supercomputing capacity to try to understand and manage that data. We're now in the third gen, or the, I'm not sure which gen they call it now, but it's moving so fast, the technology, that we can start to sequence base pairs and align them in real time. We were most often or commonly using MinION right now. It's this tiny, looks like a thumb drive. You put your sample on it, you plug it into a computer, and you can see in real time sequences starting to align. Now, you'll get a lot of messy data there, a lot of sequence data that is, it's unknown, it doesn't map to anything, but where we really can make inferences or conclusions are when we get a lot of sequences that overlap. Then you can get this huge depth and coverage which gives us a lot of confidence in that particular region. And for viruses, when you get a lot of overlapping sequences for a known virus or related virus family, that's when we can make conclusions about it. So there's a lot of methods, optimization, sample processing, data analytics. One of the hardest positions to hire in this field right now is a bioinformatician. It is so hard to get somebody who can handle these types of data, build pipelines to analyze and understand it. That is outside my expertise. I work with the veterinary school. They've got a great team there that does this. And my go-to colleague, he just left for South Dakota State University, and I still partner with him a lot to do the bioinformatic work. But I get the samples, I can sequence it and then give it to them to analyze it, and then we get the outputs to understand it. Hopefully, Colleen, that clarifies it. Happy to chat more if you want to know about the process or the people. And I'm standing between you and the pizza, so are we good? All right. Thanks, everybody.